Mauro D'Amato. I'm a professor of medical genetics in Casamassima at the University LUM in the south of Italy, and I'm also an Ikerbasque research professor in Sicbiogune in Bilbao, Spain. So this is a very good question. Actually, there's quite a few different definitions of IBS. I can give you the IBS definition that we have used in this study, which is following the Rome criteria. So now the Rome criteria are criteria that have been put together by a panel of experts in the area of neurogastroenterology. And the idea is to define, in an accurate and possibly useful way what IBS is. So now, according to the Rome criteria, IBS is abdominal pain and discomfort, because this is the third version of the Rome criteria that we've used, associated with a change in bowel habits. This could be a change in the number of bowel movements, or, for example, in the consistency of the stool. And so these are definitions that are, that serve the purpose to characterize and identify a certain subgroup of patients. Not necessarily these are the same definitions that everyone is using. Possibly they should use this, but not everyone is using, for example, in the clinical routine. And one of the major challenges to study IBS is, in fact, starting already with the definition that is used. We know from genetic studies that we've carried out using different definitions that we can see slightly different results in terms of what are the genes involved, depending on the definition that we use. This is one of several conditions that are referred to as disorders of gut-brain interactions. We don't really know well what is the problem here. We don't know what's causing these conditions. And we also don't know what is the reason why some people respond to treatment and others don't. Irritable bowel syndrome, so IBS in particular, is characterized by a number of symptoms as a complex array of symptoms, which is including constipation, diarrhea, bloating, abdominal pain, and so on. And here, one factor which is special, if you want, about irritable bowel syndrome is that women are much more affected than men. We also, also in this case, we don't know what is the reason, possibly because of some, the involvement of sex hormones and things like that. Usually, this condition has been seen as not that serious. It's only recently that people have started to investigate a little more in terms of the pathophysiology behind it. But again, I can tell you that the quality of life of patients is severely affected. This is the first reason why people refer to a gastroenterologist, and in terms of cost to society is up to 1% of the healthcare budget annually. So it's a very important condition, affects many people, up to 10% of the general population, and there's no treatment that works for everyone. So with the genetics that we are investigating, we hope we can at least identify some of the mechanisms that may translate into novel targets for therapy.
My name is Leticia Tavares, and I'm a Postdoctoral Research Fellow at the Hypertension Research Laboratory within the School of Biological Sciences of Monash University. We aim at investigating the genetic underpinnings of IBS by defining it according to the gold standard Rome criteria, which classifies IBS based on a collection of patients' reported symptoms. And for this, we conducted what we call a genome-wide association study, also known as GWAS, which is a high-throughput method used to compare the DNA profile across the entire human genome between individuals with and without a phenotype of interest, in our case, IBS.
This is a study that started more than 2 years ago. This is the time when I was at Monash University in Melbourne. And in fact, the lead author is Leticia Camargo Tavares, was one of my PhD students. She's now a postdoc in the Hypertension Lab of Dr. Marquez. So Monash has been involved. My current institutions, LUM University in Italy and Sicbiogune in Bilbao, have also been involved. And then there are collaborators from the University Medical Center Groningen who have been working on data from the Netherlands. In particular, we have been using two large data sets from two population-based cohorts. These are the UK Biobank cohort from England and the Lifelines cohort from the Netherlands. These are very important resources for the scientific community. Population-based cohorts that have a lot of different layers of data available to scientists. And this is what we did. We applied for access to this data with our idea to study the genetics of IBS. And so big thanks to these resources that allow researchers like us to study data that are not produced in-house.
The study encompassed more than 100,000 individuals, which were identified from 2 large cohorts named UK Biobank and Lifelines. We could identify more than 24,000 patients with IBS, and we compared their genetics with more than 77,000 symptom-free individuals. In both cohorts, they encompassed anonymized genetic, lifestyle, and detailed information about the gastrointestinal health of these participants, which made them very suitable, the most suitable to study IBS in large scale.
This is not the first time that irritable bowel syndrome is linked to dysmotility. We are the first to demonstrate, if you want, a genetic link between these two conditions, but there's a couple of papers, epidemiologic studies, where they have already linked irritable bowel syndrome and a number of symptoms like constipation to cardiovascular conditions, hypertension, and so on. What is the novelty here that we demonstrated in this study is that they have a partial common genetic basis. So there are some genetic factors that in fact predispose to both of them. This is important because if it's so, and we can confirm this in follow-up studies, there may be possibilities to use drugs and treatments that are used for one or the other, and in fact, it could be used for both. There's already at least one example of this in the literature. This is a study from the Mayo Clinic in US, to which we also contributed, where an ion channel that is important in arrhythmias, in like the Brugada syndrome. These are cardiovascular conditions where the heartbeat is not correct. This gene in particular, which is an ion channel, has also been shown to be important for the peristalsis. And so that's what controls really the movement of your intestinal tract. So now what is remarkable is that in a pilot study, this is a proof of principle study, a drug which is called mexiletine, and it's used for arrhythmias, has also been shown to be relevant and, in fact, to work to relieve constipation in at least one patient. So there are some examples already in the literature where some drugs and therapeutic approaches that are used for conditions like hypertension, cardiovascular disease might be, in fact, relevant and potentially usable also to treat IBS patients.
Our study revealed 4 genomic regions where certain DNA changes were more common in IBS patients than in controls and were then associated with a higher susceptibility to IBS. And 2 of these risk loci, they are novel, they have never been previously reported. And interestingly, within these genomic regions, there are genes that are implicated in functions such as the control of our circadian rhythm, the control of the intestinal mucosa permeability, and also gastrointestinal motility, which are all biological mechanisms that are plausible to be part of the causes of IBS. So these findings are very exciting because they provide a novel insight into the pathophysiology of IBS, and they also highlight some biomarkers that can be used as potential drug targets, warranting further investigation in follow-up studies. In addition, our study revealed that IBS heritability, which is basically how genes determine the risk to disease, is higher than previously thought. So this might stem from the fact that we adopted a more standardized structure classification of IBS according to the Rome criteria as mentioned.